Research activities in the Jacobson Group range from instrumentation development in fundamental studies of micro and nanofluidics, charge transport, and optofluidics, to biological assays, including virus assembly, bacterial development, cancer diagnostics, and phylogenetic toxicology. Group members will now give a brief overview of their projects. Hi, my name is Mike. I'm a fifth-year graduate student in Dr. Jacobson's lab, and I work on the nanofluidic project. What I look at is the detection of HPV capsid, and our main technique is called resistive pulse sensing. How it works is we have a capsid that'll get electrophoretically driven through this electrically biased pore. Here we have the capsid is not in the pores, and we get a baseline current of approximately 17 nanoamps. As the particle is driven into the pore, we get a displacement of electrolyte, which causes an increase in resistance and so a decrease in the current amplitude. And from the difference from this baseline to the trough here, we can get a sense of how large that particle is. We have 3 pulses, and that's so that we can have improved signal averaging from 3 pores. And here you can see we can have hundreds of particles in a matter of minutes, each of which is measured 3 times.
Hi everybody, my name is Dee, and this is the focused beam instrument that we use to make our nanofluidic device. So here's the middle column is the one we use to make scan electron microscopy to help us find our samples. And back here we can control a very focused beam of gallium arcs, which can sputter the materials of our substrate surface and to make the patterns that we design. And then we can also do depositions with this gas injection system. So with that, we are able to make various nanostructures.
Hello, I'm Tanner, and I'm a second-year analytical student.
Hi, I'm Lizzie, and I'm a first-year analytical student. We both are on the nanofluidics project with an emphasis on extracellular vesicles.
Extracellular vesicles range anywhere from 30 nanometers to a few microns in diameter.
It is important to study EVs because they have been shown to resemble the mother cell from which they come from. They have also been linked to cell-to-cell communication, and they can be used as biomarkers to identify diseases.
We also utilize resistive pulse sensing, where we use this technique to characterize single particles for their size and their charge.
I work on the separations project, and here you can see that we use the focused ion beam in order to mill a device to perform these separations of polystyrene beads. Here's a live video of this separation occurring. This will later be adapted to research EVs.
Hello, my name is Namyu Lee in Professor Jacobson's Group, and I'm a first-year student doing glycan project. So the goal of my project is to identify potential glycan cancer biomarker from body fluids. I use the samples such as human serum exosomes, human urine, and human serum to get glycan. For my project, to analyze the data, I use CEMS, the capillary electrophoresis mass spec. And here's the brief data of the glycan profile obtained from Dr. Warren Song. And the key points of this technique is we can separate the isomers of the glycan profile, like this tube.
I'm GU, a fourth-year grad student in Jacobson's lab. So this instrument is our detection instrument for our glycans. It's called Orbitrap Fusion Lumos, a very advanced instrument developed by future scientists. So with this instrument, we can detect very small molecules with very high resolution. It helped us to achieve what we wanted for the cancer screening and also for our phylotox project. So the phylotox project will focus on some molecules, include lipids, sugar, and amino acids, and we'll talk about that in the next chapter. In phylotox project, our goal is to profile the metabolism of model organisms, and also we'll examine the metabolomic changes under regulatory exposure, which are the small molecules I mentioned previously. In this side, you can see this is a phylogenetic tree. Our targeted model organism is going to include C. elegans, fur flies, zebrafish, Xenopus, and probably in the future, we'll also use human cell lines for our future project. And the techniques we're going to use here will be nano-electron spray ionization direct infusion mass spec.
Hi, I'm Gerardo. I'm a first-year student, and I work with CU in the tandem mass spectrometry focus of the Phylotox project. We use the Traversa Nanomate and the Orbitrap Fusion Loomis for automated direct infusion mass spectrometry. The Nanomate acts as an autosampler and an ion source by using this electrospray chip. By using direct infusion instead of chromatography, we're able to run our experiments in a matter of seconds instead of hours. And it's worth noting that the Fusion Loomis and the Nanomate were not designed to work together. So the machine shop, the electronic shop, and the Laboratory for Biological Mass Spectrometry facility here at IU all work together to create the setup.
I'm Brigham Pope. I'm a second-year graduate student in the Jacobson Group. I'm in the photolithographic mapping project. After we mill nano apertures into thin layers of metal, we illuminate them with UV light to get a 3D model of how the light passes through. We use a negative tone photoresist that lets us see exactly where the UV light has passed through these apertures. And by seeing these three-dimensional models, we can determine what uses we can use these apertures for in microfluidic devices.
Hi, I'm Lara and I'm a second-year grad student here at the Jacobson Lab. I work in the bacteria project where we use this microfluidic device to trap and image bacteria. So our device works with vacuum and pressure to control the different valves and control the fluid as well as the bacteria grow in the channels and the delivery of the media. We here have our microscope in which we use fluorescence to track the different bacteria. So over here in our video, you can see how across time we are able to see how the bacteria grow in our channels and track different proteins inside the bacteria.